So, dear students, as you know already, concerned with my side portion, previously we have come across 4 chapters. Number 1, reproduction in organism. Second, sexual reproduction in flowering plant. Third is human reproduction. Fourth is reproductive health. Followed by that, so today I plan to switch over to next one more important chapter, Biotech. So today I plan to switch over to Biotech. Actually, it is a short form of biological technology. It is a short form of biological technology. Biotech is a short form of biological technology. As the name itself indicates, it is the technology of biological origin. Look over here. So today I plan to switch over to a very important chapter, Biotech. It is a short form of biological technology. Biotech is a short form of biological technology. As the name itself indicates, it is the technology of biological origin. As the name itself indicates, it is the technology of biological origin. I will provide you technology of biological origin, making curd, bread, alcohol, wine, making curd, bread, alcohol, wine. All they are microbe mediated processes. Making curd, bread, alcohol, wine. All they are microbe mediated processes. All they are included under biotech. Look over here. Biotech is a short form of biological technology. As the name itself indicates, it is the technology of biological origin. I will provide you few examples for technology of biological origin, making curd, bread, alcohol, wine. All they are microbe mediated processes. All they are included under biotech. In addition to that, it also includes IVF and ET. IVF and ET. You know already, in vitro fertilization and embryo transfer. It is commonly called as test tube baby. It is commonly called as test tube baby. IVF and ET we have come across in detail in reproductive health chapter. It includes test tube baby, synthesizing a gene and using it. Synthesizing a gene and using it. You know already gene, it is the fundamental unit of heredity. Gene is the fundamental unit of heredity associated with a particular character. Gene is the fundamental unit of heredity associated with a particular character. Synthesizing gene and using it. Developing DNA vaccines. DNA vaccines. Developing DNA vaccines. You know already DNA, it is commonly called as master molecule in the biological system. DNA is commonly called as master molecule in the biological system because it will control all the activity of the cell directly or indirectly. DNA vaccines and correcting a defective gene. Correcting a defective gene. So correcting a defective gene is nothing but gene therapy. Look over here. Making curd, bread, alcohol, wine. All they are microbe mediated processes. All they are included under biotech. In addition to that, it also includes test tube baby, or you can call it as a in vitro fertilization and embryo transfer, synthesizing a gene and using it, developing DNA vaccines and correcting the defective gene. Correcting the defective gene is nothing but gene therapy. You know gene, it is the fundamental unit of heredity associated with a particular character. Replacement of the faulty gene by a normal functional gene is called as gene therapy. Replacement of the faulty gene by a normal functional gene is called as gene therapy. But nowadays or in modern days, same thing is achieved in modern days. Same thing is achieved by using genetically modified organisms. So in modern days, same thing is achieved in a large scale by using genetically modified organism. Now question arises, what is this genetically modified organism? An organism whose genes have been altered, an organism whose genes have been altered by manipulation is called as genetically modified organism. So in modern days, same thing is achieved in a large scale by using genetically modified organism. An organism whose genes have been altered, an organism whose genes have been altered by manipulation is called as genetically modified organism. When they arise question, define biotech, don't write it is the technology of biological origin. This meaning will not carry mark. So I will introduce 3 beautiful definitions, but you have to select the only one which they have mentioned in the NCERT book. So now look over definition number 1. I will provide you 3 beautiful definitions. As already I requested you to remember the one which they have put forward in the NCERT syllabus. Okay, definition number 1. It is the applications of the principles of life, not principles. It is the applications of the principles of the life and the technologies. It is the applications of the principles of the life and technologies to produce human wanted substances. Look over this one very simple definition. It is the applications of the principles of life. It is the applications of the principles of the life and the technologies to produce human wanted substances. Now question arises, what is this human wanted substances? It includes antibiotics, enzymes, hormones, DNA vaccines and blood clotting factors. Antibiotics, you know, they will fight against the antigens. So detailed story is there in the chapter. Later we will come to know this matter under the guidance of Sir. Antibiotics will fight against antigens. Enzymes and hormones already once I covered. Back to PUC first year, chapter Structural Organization in Animals, where I come across glands are classified into 2 groups. Glands are of 2 groups, exocrine glands and endocrine glands. Glands with duct is called as exocrine glands. Glands with duct is called as exocrine glands, and glands without duct is called as endocrine glands. Secretion of the exocrine gland is called as enzymes, and the secretion of the endocrine gland is called as hormones. You know already, glands with duct is called as exocrine glands. Glands without duct is called as endocrine glands. Secretion of the exocrine gland is called as enzymes. Secretion of the endocrine gland is called as hormones. You know, hormones are commonly called as chemical messengers. But whereas enzymes are biocatalyst which accelerates the biochemical reactions. Enzymes are biocatalyst which accelerates the biochemical reactions. DNA vaccines and blood clotting factor. I'll provide you a little idea related to blood clotting factor. Back to principles of inheritance and variation, where I come across hemophilia and color blindness. Hemophilia and color blindness will follow zigzag inheritance or crisscross inheritance or skip generation inheritance. Hemophilia and color blindness will follow zigzag inheritance or criss-cross inheritance or skip generation inheritance, because the character which is present in the grandfather is going to appear in the grandson through daughter. You know, hemophilia is also called as royal disease because it is noticed in the royal families of the Russia and England. Queen Victoria is a carrier of this disease. It is also called as bleeder's disease because the blood will oozes out continuously. Even from the slight cut ends. So this blood clotting factor will help hemophilic patients. Blood clotting factor will help hemophilic patients. Okay, I hope that first definition is clear. Biotechnology is the applications of the principles of life and technologies to produce human wanted substances. So this human wanted substances includes antibiotics, enzymes, hormones, DNA vaccine and blood clotting factors. Now look over the next definition. Now look over second definition. Biotech deals with the technique of using living organisms. Biotech deals with the technique of using living organisms or enzymes from the organisms to produce products and processes. Look over second definition. Biotech deals with the technique of using living organisms or enzymes from organisms to produce products and processes. Our technique of using living organisms. Living organisms includes both plants and animals. In that also both prokaryotic and eukaryotes. Both prokaryotes and eukaryotes are enzymes from organisms to produce products. Products includes antibiotics, enzymes, hormones, vaccines, blood clotting factors, etc. Processes. I'll provide you one simple example for the process. Back to PUC first year, chapter respiration in plants. Respiration in plants, where we have come across process of respiration, the process of respiration which takes place in presence of the process of respiration which takes place in presence of oxygen is called as aerobic respiration. The process of respiration which takes place in presence of oxygen is called as aerobic respiration. But the process of respiration which takes place in absence of oxygen is called as anaerobic respiration. Under anaerobic respiration, we have come across fermentation. Two types of fermentation we have come across. One is alcoholic fermentation, another one is lactic acid fermentation. For CET purpose, please remember, in lactic acid fermentation, carbon dioxide is not produced. Okay, I hope that the point is clear. Products are listed already. Process, I'll provide you one example. It is fermentation. The process of respiration which takes place in absence of oxygen is called as anaerobic respiration. Under that, you have come across two types of fermentation. One is alcoholic fermentation, where C2H5OH is produced or ethyl alcohol is produced. In lactic acid fermentation, lactic acid is produced instead of ethyl alcohol from pyruvic acid or pyruvate. For CET purpose, please remember, in lactic acid fermentation, carbon dioxide is not released. Okay, so I hope that second definition is also clear. Next, I will switch over to one more definition. Look over next one more third definition. Okay, look over this third definition. As already I mentioned, have to remember the definition which they have quoted in the new NCERT book. According to European Federation of Biotechnology, according to European Federation of Biotechnology, it is an integration of natural science. It is an integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Like this, you cannot follow the meaning. Look over carefully. According to European Federation of Biotechnology, according to European Federation of Biotechnology, okay, it is an integration of, integration means combined, integration means combined, natural science. Natural science includes physics, chemistry, and biology. Korea. Integration means combined of natural science. Natural science includes physics, chemistry, biology, and organisms. You know already organisms. Cells, as you have come across in first year. Cell is the structural and functional unit of life. Cell is the structural and functional unit of life. Parts thereof and molecular analogous. Molecular analogous means you are producing the structure similar to that of the DNA and RNA. As already we have come across DNA and RNA in detail in molecular basis of inheritance. Molecular analogous means you are producing the structure similar to that of DNA and RNA. For products, already you have come across many products and services. So out of 4 definitions, better you remember the third one. Out of 4 definitions, better you remember the third definition. Okay, according to European Federation of Biotechnology, it is an integration of natural science and organisms, cells, parts thereof, and molecular analogous for products and services. Next is principles of biotechnology. So now look over, dear students, principles of biotechnology. The principles of biotechnology. It includes genetic engineering and bioprocess engineering. So look over principles of biotechnology. One is genetic engineering, second is bioprocess engineering. Genetic engineering involves in vitro transfer of sequences of DNA in vitro transfer of sequence of DNA from one organism to another organism, from one organism to another organism, from one organism to another organism to change phenotype, the phenotype of the host organism. Look over here. The two principles of biotechnology are genetic engineering and bioprocess engineering. In vitro transfer of sequence of DNA, it involves in vitro. In vitro means outside the body of the organism, or you can call it as in test tubes. In vitro transfer of sequence of DNA, sequence means a small piece of DNA, from one organism to another organism to change the phenotype of the host organism. Look over here. It involves in vitro transfer of sequence of DNA. Sequence means a small piece of DNA from one organism to another organism to change phenotype of the host organism. Back to genetics, where I come across phenotype and genotype. Genotype means genetic makeup of an individual. Genotype means genetic makeup of an individual. Phenotype, externally expressed characters. So two technical terms I come across in genetics: phenotype and genotype. Genotype, genetic makeup of an individual. Phenotype, externally expressed characters. Externally expressed characters. So I hope that point is clear. Genetic engineering involves in vitro transfer of sequence of DNA, a piece of DNA, from one organism to another organism to change the phenotype of the host. Genotype, genetic makeup of an individual. Phenotype, externally expressed character. Okay, phenotype, externally expressed one. Now, genetic engineering involves recombinant DNA, otherwise I will write here. It includes recombinant DNA, gene cloning, and gene transfer. Look over here. So this genetic engineering includes recombinant DNA technology, gene cloning, and gene transfer. It includes recombinant DNA, gene cloning, and gene transfer. If you want to understand the next para, try to recollect the matter which you have come across in chapter number 1. So back to chapter number 1, reproduction in organism, where we have come across reproduction are of two types, asexual and sexual. Production of an offspring by a single parent, production of an offspring by a single parent without formation and fusion of gamete is called as asexual reproduction. So dear students, now move back to first chapter, reproduction in organism, where we have come across definition of reproduction and two types of reproduction. Reproduction are broadly classified into two groups, asexual and sexual. Production of an offspring by a single parent without formation and fusion of gamete is called as asexual reproduction. The individual which are produced through asexual reproduction is almost identical to each other. Hence the term clone is used to describe such an individuals which are morphologically and genetically similar. The term clone is used to describe such an individuals which are morphologically and genetically similar. But sexual reproduction, in sexual reproduction, variations are created. It involves the formation of male and female gamete either by the same individual or different individuals of opposite sex. I hope that the two meanings are clear. Now move to this side, one more para I would like to introduce. During sexual reproduction, variations are created. During sexual reproduction, variations are created. During sexual reproduction, variations are created. These variations may be useful. Such variations are not produced during asexual reproduction. So look over here, dear students. During sexual reproduction, variations are produced. Some variations are useful. Such variations are not produced during asexual reproduction. In hybridization experiment, both desired and undesired. During hybridization experiment, both desired and undesired genes are transferred to the offspring. Both desired and undesired genes are multiplied in the hybrid, but genetic engineering will enable to transfer only the desired DNA to the target organisms. Look over here. During sexual reproduction, variations are created. Some of the variations are useful. These variations are not created during asexual reproduction. In hybridization experiment, both desired and undesired genes are multiplied in the hybrid. But genetic engineering will enable to transfer only desired DNA to the target organism. Look over here. Genetic engineering will enable to transfer only the desired DNA to the target organisms. So don't bother about all this recombinant DNA, gene cloning and gene transfer. In later classes, you will come to know this matter. Next is bioprocess engineering. Bioprocess engineering. Bioprocess engineering will enable maintenance of sterile environment, will enable the growth of desired microbes or eukaryotic cells in a large quantity for the production of human wanted substances. Look over here. Bioprocess engineering. It will enable the growth of only desired microbes or eukaryotic cell in a large scale to produce human wanted substances. Maintenance of sterile environment. Sterile means free from microorganism. Bioprocess engineering. Maintenance of sterile environment. Sterile means free from microorganism. What word sterile you have come across in reproductive health is different. Sterile means unable to conceive. But here sterile means free from microorganism or microscopic organisms or microbes. You know, microscopic means they are not visible to the naked eyes. It will enable the growth of only desired microbes. Desired means required microbes or eukaryotic cell in a large scale to produce human wanted substances. Already I put forward a list of human wanted substances. Sterile means free from microorganism. It will enable the growth of only desired microbes. Microbes means which are not visible to the naked eyes. So they can be seen only with the help of microscope. Or eukaryotic cell, you know already, U means true, or well, karyon means nucleus. In eukaryotes, nucleus is well developed and they have cellular bond organelles. Eukaryotic cell in a large scale to produce human wanted substances. This human wanted substance includes enzymes, hormones, antibiotics, DNA vaccines, etc. That ends introduction. Now look over, dear students, tools of recombinant DNA. Tools which are used in genetic engineering, or you can call it as a recombinant DNA technology. Tools which are used in recombinant DNA technology or genetic engineering. Actually, many tools are there, but for your syllabus, they are quoted 3: enzymes, vectors, host, and indirectly they are mentioned bioreactor, or you can call it as a fermenter. So the tools which are used in genetic engineering or recombinant DNA technology are enzymes, vectors, host cell, and bioreactors. Now first look over enzymes. There are 3 enzymes that are quoted in your syllabus, but the last one, polymerase, you have come across in detail in molecular basis of inheritance. Polymerase enzyme, already we have come across in detail in molecular basis of inheritance, but in this chapter you are going to study more related to restriction enzymes. You are going to study detailed matter related to restriction enzymes. Restriction enzymes will come under a large class of enzyme known as nucleases. Restriction enzymes will come under a large class of enzyme known as nucleases. They are of 2 types: exonucleases and endonucleases. Look over here. Restriction enzymes will come under a large class of enzyme known as nucleases. Restriction enzymes will come under a large class of enzyme known as nucleases. They are of 2 groups: exonucleases and endonucleases. Exonucleases and endonucleases. Exonucleases will remove nucleotides. It will remove the nucleotides from the end of DNA. Exonucleases will remove the nucleotides from the end of DNA. I hope that you have remembered nucleotide is equal to nucleosides plus phosphate. Okay, so look over here. Restriction enzymes will come under a large class of enzyme known as nucleases. Nucleases are of 2 types: exo and endo. Exonucleases will remove the nucleotides. They will remove the nucleotides from the end of DNA. Nucleotide is equal to nucleosides plus phosphate. Back to molecular basis of inheritance, where I come across nucleotide is equal to nucleosides plus phosphate. Nucleoside is equal to nitrogenous bases plus pentose sugar. Okay. Second is endonucleases. They will cut the DNA. Endonucleases will cut the DNA. Now next matter. Steward, Lin and Werner Arber. Steward, Lin and Werner Arber isolated 2 enzymes. Which restricted the growth of bacteriophages in E. coli bacterium. Look over here. Stewart Lin and Werner Erber has isolated two enzymes which restricted the growth of bacteriophages in E. coli bacterium. One will add methyl group Stewart Lin and Werner Erber isolated two enzymes which restricted the growth of bacteriophages in E. coli bacterium. One will add methyl group to the DNA, one will add methyl group CH3, methyl group to the DNA, and another one will cut the DNA. So I hope that you have remembered bacteriophages and E. coli. Back to first year. Viruses are classified based on different criteria. Viruses are classified based on different criteria. One is the type of cell which they infect, plant viruses. Viruses which infect the plant are called as plant viruses. Viruses which infect the animal are called as animal viruses. Viruses which infect the bacteria are called as bacterial viruses or bacteriophages. Back to first year. Try to recollect the matter. Actually, viruses are classified based on different criteria. One is based on the type of the cell which they infect. Plant viruses. Viruses which infect the plant cell are called as plant viruses. Tobacco mosaic virus. Viruses which infect the animal cell are called as animal viruses. For example, HIV. Viruses which infect the bacteria are called as bacterial viruses. A classic example which we come across in first year is T4 bacteriophage. T4 bacteriophage. Okay. E. coli is an bacteria. Escherichia coli. Okay. I hope that this first para is clear. Stewart Lin and Werner Erber has extracted two enzymes or isolated two enzymes which restricted the growth. Restricted means they will prevent the growth of bacteriophages in E. coli bacteria. One will add methyl group to the DNA. One will add methyl group to the DNA and another one will cut the DNA. The one which cuts the DNA is called as restriction endonucleases. Restriction endonucleases. Korea. One will add methyl group to the DNA and another one will cut the DNA. The one which cuts the DNA is called as restriction endonucleases, or you can call it as a natural scalpel or molecular knives. The one which cuts the DNA is called as restriction endonucleases. It is also called as molecular scissors, or molecular knives or natural scalpel. Okay, I hope that first point is clear. Okay.